terms of peers, it basically consists of rectum, the large intestine, the small intestine, the pancreas, the stomach, also called gaster, and the liver with the gallbladder. The esophagus is also part of the system as well as various salivary glands near the mouth. First, the food is broken up in the mouth by the teeth and then mixed with saliva with the help of salivary glands. Saliva contains a digestive enzyme called amylase that already begins to digest carbohydrate in the mouth. It splits carbohydrate into smaller units. The ball-like mixture of food with saliva, also known as bolus, is pushed into throat by the tongue and finally into the esophagus, which propels the bolus to the stomach. The esophageal lumen, that is the opening inside the esophagus, is very flexible, which allow bolus of different sizes to be transported. The esophageal consists of several layers. These layers occur throughout the entire digestive tract. The two outer muscle layers are responsible for perisses. Through these two muscles, the bolus can be transported from the mouth to the stomach, even if the person is standing on his head. The stomach is often divided into six parts. The stomach, composed of a similar structure to the esophagus, it is a longitudinal muscle layer on this side. But underneath, we can find circular muscle fibers. In addition to this, there is an oblique muscle layer overlaying the mucus. On the inside, there are rugulae that allow the stomach to enlarge when the food is consumed. The stomach wall contains gastric glands. They produce mucus which is able to protect the stomach wall from the secreted gastric acid. The gastric acid is produced by simply smelling or seeing the food, but also spices and the stretching of the stomach causes the secretion. That is the release of gastric acid. About 1 or 2 liters of gastric acids are produced per liter. Since the esophagus do not have protective mucus layer like stomach and esophagus are separated by sphincter. It relaxes when a bolus is pushed down from the esophagus into the stomach and then contracts to prevent acid and the food going back up. The gastric juice consists among other things of hydrochloric acid and enzyme pepsin, the intrinsic factor and lipids of the digestion for fat. In addition to nutrients, food also contains bacteria that can damage the body. The components of hydrochloric acid are able to destroy harmful bacteria. In addition, hydrochloric acid prevents pepsogen and also released by the gastric glands into the pepsin. Pepsin is able to break down proteins in the stomach. For vitamin B12 absorption in the small intestine, the intrinsic factor is needed, which is produced by gastric glands. The vitamin must combine with intrinsic factor, then it can be absorbed later by the small intestine. Vitamin B12 helps keep the body nerves and blood cells healthy and helps to make DNA. It also contains gastric lipids and acid resistant enzyme for fat digestion. In the stomach, the gastric lab splits the triglycerides into free fatty acid and a digestion whereby only the free fatty acid can be absorbed by the body. More effective fat digestion takes place in small intestine through gastric juice and stomach movements which take place approximately every 20 seconds. The individual boluses are mixed to a semi-fluid mass of partially digested food, the so-called scam. The scam cannot enter the duodenum at first because there is a snipture at the stomach exit. The pyloric snipture resembles the esophageal snipture. The pyloric snipture opens only a few millimeters so that larger pieces remain inside the stomach. In the first section of small intestine, the duodenum, bile, and the pancreatic secretions are mixed with the sym via the ampulla of Vader. Pancreatic juice contains number of digestive proenzymes and enzymes. In order for these to do their job, a higher pH value than that in the stomach is necessary. For this reason, pancreatic juice contains sodium hydrogen carbonate. Hydrogen carbonate is able to neutralize the acid in the scam and thus produce optimum pH value of 7 or 8. Pancreatic juice also contains proenzyme. It is only through electrolysis released by the duodenum wall. That is proenzyme trypsin becomes trypsin which can split proteins and activate other trypsin genes. We also find alpha amylase which we had already found in the mouth. It now does the rest regarding the splitting of carbohydrate which it converts into maltose and amylose. Furthermore, pancreatic lipase is able to split triglycerides into two free fatty acids. The glyceric lipase, as we have seen before, can produce only one free fatty acid. The pancreatic lipase can cleave triglycerides excellently because the bile breaks down the fat into tiny droplets. This is called emulsification. Numbers of other enzymes are part of the pancreatic juice, but these will not be explained in detail here. Bile is produced by liver cells and transported to the gallbladder. The bile is stored in the gallbladder and finally added to food in the duodenum via alpha of Vadar. The pancreas also releases juice via the alpha of Vadar. The small intestine consists of three sections: duodenum, jejunum, and ileum. The ileum continues into the large intestine in the right lower abdomen. The duodenum and the jejunum have circular folds to increase and contact surface with the food. These circular folds extend about 1 centimeter into the lumen in the small intestine. These folds are converted with small finger-like projection called villi. Villi increase the surface considerably. Each villi is about 1 millimeter long. Each villus contains blood capillaries and a implicating capillary called lectal, which we will see later. The nutrients marked green here are absorbed by the villus and transferred to the blood capillaries. Some nutrients, such as glucose, do not require a carrier. They are transported freely in the bloodstream. Other nutrients, such as iron, requires transport Proteins like ferrin. Fats are transported by climacrons, which are lipoproteins. The triglycerides to be transported are virtually enclosed in lipoprotein. The climacrons and triglycerides are then transported to the lacteal of the villi. Each villus is covered by even smaller microvilli. They multiply the initial surface considerably. The microvilli absorb nutrients and transport them to inside. The last part of small intestine is lumen. It does not have, unlike the duodenum and jejunum, circular folds. The lumen absorbs electrolytes such as calcium for building bones, hairs, and teeth. Trace elements such as zinc for the sperm production and immune system, the vitamins such as B12 for the information and maturation of red blood cells, and remaining bile acid which is transported back to the liver via bloodstream. As with the esophagus, the food is transported by peristalsis. In contraction similar to the mixed sign, which is shown here yellow and red, to make it easier to see the mixing process. The large intestine is thicker than the small intestine. It is about 1 meter long and surrounds the small intestine. The small intestine is connected to the large intestine via Böhm's valves. It opens when the serum is to pass through the small intestine to the large intestine. The large intestine does not have any villi like the small intestine because most digestible substances have been already absorbed in the small intestine. However, the large intestine has an estimated 100 billion bacteria inside. They are important for many other tasks such as production of vitamins and the decomposition of fibers for the body's energy production. Many of these bacteria are important part of the immune system by killing harmful germs to the peristalsis. The cycle is transported from the ascending colon to the transverse colon to the ascending colon. On its way through the large intestine, water is removed from the scan. Furthermore, mucus is added for proper secretion of waste, substances that cannot be absorbed through the small intestine or the large intestine remain in the rectum and finally excreted through the anus.